One of the most controversial science stories of the year involves stem cells, cells that may one day cure terrible diseases. But the problem with stem cells is they often come from embryos, thus the phrase embryonic stem cells. And embryos, while they start no bigger than a grain of sand, can become babies. So there's a moral issue here. But what if, and we're just going to think out loud, what if you could make an embryonic stem cell without creating an embryo? Could you? Should you? Well, we know someone who's trying. Reporter Julia Court has the story.
As he winds through the labs of MIT, Alex Meissner is trying to sidestep an ethical minefield. He hopes the tiny cells that float in his petri dish might hold the key to solving one of the biggest controversies facing science. Whether it's ethical to use cells from human embryos in medical research.
I believe this research has enormous potential, and it will revolutionize medicine in the years to come.
I don't want to live in a world, I don't think anyone wants to live in a world where we're destroying embryos, we're creating them and destroying them in mass quantities for research purposes.
While experts debated here, halfway around the world, South Korea seemed to be forging ahead. Last August, they claimed a spectacular first. A cloned dog. But since then, the Korean king of clones, Hwang Woo-suk, has been dethroned, accused of fabricating his results, including groundbreaking work cloning human embryonic stem cells. The Korean fiasco has cast a cloud over the field, but U.S. researchers still want to move forward, believing embryonic stem cells hold great potential for medicine, since they can become any kind of cell in the human body. But because they come from early human embryos, funding for the research is extremely limited.
Unfortunately, here in the United States, even though we're chomping at the bit to do the work, the kind of funding restrictions that we're under, the kind of presidential policy that we're under, is really the hindrance.
Physician and researcher George Daly is eager to create stem cells that are an exact match to sick patients. To do that, he wants to try cloning technology, but so far, the only cloned cells he's made belong to mice.
We're essentially frozen in time at 2001, while the rest of the world has moved forward very, very aggressively.
The controversial cloning process would work like this. Take an egg from a woman's ovary, remove its genes or DNA. Then get a cell, like a skin cell from a patient, and put its DNA into the egg. The egg is stimulated to divide, as if it had been fertilized. After a few days, the early embryo has 50 to 200 cells. The goal is to turn some of these into embryonic stem cells that would be a perfect genetic match to a sick patient. But because the cloned embryo would be destroyed in the process, critics say this research is wrong. With the destruction of human embryos at the center of the debate, a few scientists approached the new year asking if it might be possible to make both sides happy. What if they could find a way to create embryonic stem cells without destroying any embryos?
I think it should be doable.
Physician and ethicist William Hurlbut thinks the solution may lie in tinkering with the cloning process.
There's a way to get the cells without creating embryos. We just have to have the constructive conversation to find the way.
The challenge intrigued MIT's Rudolf Janisch, a leading expert on stem cells and cloning. With the help of graduate student Alex Meissner, Janisch tried to create something that would yield stem cells, but was not an embryo at all.
What is an embryo? What's important? I think an important part of the sense of an embryo is the potential to develop, to organize itself.
The Janisch team set out to eliminate that potential. First, they took a tiny bit of skin from a mouse's tail and let it grow in culture. With the skin cells growing in a petri dish, Meissner concocted a special virus and added it to the skin cells. The virus went to work and inserted an extra bit of DNA into the skin cell DNA. Meissner performed the cloning, sucking up a skin cell now containing the extra DNA and transferring it into a mouse egg. And then the team waited and watched. Janish's hope was that the extra bit of DNA they'd added would shut down one key gene, a gene that's crucial for organizing the early mouse embryo and triggering the construction of the placenta.
The placenta, of course, is very important in signaling the fetus to develop. Without the placenta, it cannot. And the experiment worked very straightforward. It was actually surprisingly simple to us.
With that key gene turned off, the unorganized cluster of cells did not develop normally, and Janisch says would never become a baby mouse. But it could be turned into useful stem cells.
The real question is, does it solve the ethical problem? Some people said yes, others said no. It's a particularly devious way to murder an embryo. I would argue from the biological point of view, it is not an embryo. It doesn't qualify to be called this. It doesn't have any potential whatsoever to ever organize itself to a fetus.
Father Thomas Berg is encouraged by Janisch's experiment.
I think the jury's still out as to whether it's an embryo or not, but I think it's incredibly interesting from a moral standpoint, from a scientific standpoint, obviously. I think what Rudi Janisch did holds out some hope that those of us who do not want to live with a future of embryo-destructive research, we might have a way out.
But so far, there is no consensus about the moral status of what Janisch created among stem cell opponents or supporters.
The scientists who play along, I think, are playing the wrong card. They think, yeah, maybe if I'm clever enough, I'll satisfy all the critics. I think the critics are not going to be satisfied. I think they don't want to see embryonic stem cell research happen, and I don't think they want to see anything involving cloning happen.
Ethical objections have slowed U.S. research. Few labs have tried to use cloning to make human stem cells, and none have succeeded. The Korean scandal means that the technique for humans is unproven. But that, for many U.S. researchers, is all the more reason to keep moving ahead.
I have patients, and I can see a way to apply this technology to study their disease and perhaps treat their disease. I want to be able to use that process today.
If we get diverted into the debate about can we make a pseudo-embryo or an embryo-like thing, you could spend the next 5-10 years trying to figure out how to tweak an egg or play with embryos. I think to spend too much time trying to respond to every possible critic is a recipe for paralysis, not for breakthroughs.